Today we're going to talk about a process called PCR. PCR is also known as polymerase chain reaction. It is a very common reaction done by researchers now that is used to amplify DNA. It was first discovered by Carrie Mullis, and now it's mainly used to look for mutations in a gene or to just sequence genes, and actually it can be used as well for restriction enzymes and a lot of different plasmid experiments as well. So PCR has three main steps. It has denaturation, annealing, and extension. And to do PCR, you need a DNA template, which can be any of your choosing. You need primers, which is also forward and backwards, so you can run it both ways and amplify the sequence together. You need your four basic nucleotides, just A, T, C, and G. There's no uracil because we're just working with DNA. You need a buffer to keep it safe, and you also need a certain type of polymerase, which is called Taq polymerase.
The first step of PCR is to denature this. So denature basically means to separate this double helix strand to the single strands. So we just use the Taq polymerase for the enzyme Taq polymerase to do that. So this double strand we need to heat for around 94 degrees Celsius for 30 seconds, somewhere around 30 seconds. And after that, the hydrogen bond gonna break it. So these two strands gonna separate to the single strands, so that the annealing can take place after that. So this is basically the denature thing, is just to break this stuff.
Annealing is the part of PCR that follows denaturation. It's a part that includes the primer. The primer is a short, about 20 base pair piece of DNA that sticks to the single-stranded DNA, and it can start at the leading or the lagging end, and it typically surrounds the target part so that can be amplified in the later step. This happens at 55 degrees Celsius, and it lasts for about 30 to 60 seconds.
Extension. The Taq polymerase extends the primer by adding complementary nucleotides. It either goes forward from 5' to 3' or backwards from 3' to 5'. This works best at 72 to 75 degrees Celsius. You're not going to find any uracil in this reaction because it's only making DNA. After extension's over, these two pieces will be separated by heat and they'll go back into the next cycle of PCR. Normally you want at least 30 cycles of PCR before you tell your reaction to stop, and by that point you have millions of copies of your target sequence.